In this video, I'll be talking about transformation, which is one of the three processes with which recombination in bacteria and viruses takes place. These three processes are conjugation, transformation, and transduction, and here I'm talking about transformation. Okay, so what is transformation? Transformation, what happens is like the bacteria or the virus will take up some DNA, and the DNA that it takes up is usually a naked DNA. What do you mean by naked DNA? Naked DNA means the DNA which is not present inside the cell, but it is present outside the cell within the intracellular matrix or within the media. Okay, and these molecules of this DNA are present in fragments, which is again taken up by the cells. The cells that are taking up are called as the recipient bacteria, and the one which are giving are called as the donor bacteria. Okay, now the transformation may be natural or artificial. Natural transformations are rare even and occurs in both gram-positive and gram-negative bacteria. So I have drawn this particular diagram to explain exactly how the process, like how does the process looks like. Okay, so I'll just start labeling. Okay, so this one is bacteria. Now, what kind of bacteria this one is? I've drawn red DNA. This red DNA is actually the DNA which is present inside this bacteria. Okay, so there is a bacterial chromosome. So this bacteria is called as the donor bacteria because this is the bacteria which will be giving this DNA. Okay, and the cells that are receiving are called as the recipient bacteria. So out here I have shown these green ones are the uh, recipient bacteria, or you can say recipient cell. Okay, then this bacteria, this donor bacteria, this bacteria are been broken down. So here what we can see is these are all the particles. Okay, so these are dyed bacteria. So basically lysis has taken place and therefore this bacteria has broken down into smaller pieces. Okay, and therefore what happens, the entire DNA will come out. Okay, so whatever DNA is present outside now within the extracellular matrix. Okay, so basically this is the extracellular matrix and this is the medium in which the cells are growing. Okay, so this particular DNA which is coming out of here, this DNA is called as fragment DNA. This is the DNA which is coming out after the cell has broken down. Okay, or we can call them as foreign DNA. We can also call them as naked DNA because again they do not have any covering, they do not have any cell, they're just outside the cell. Or this is also called as exogenous. Okay, okay. Now you can see the other cells I have drawn, some in blue and some in green. So the green ones are the recipient bacteria. That is, these are those bacteria which are competent. That means they have the ability to take a DNA. Okay, and the blue ones are the non-competent cells. Okay, these are also recipient cells, but the problem is they are non-competent. So they are non-transformants. Transformants. So basically they will not have any transformation. Okay, and the competence are they are the transformants. So basically they will have transformation after taking up the DNA. So you can see here the red part has entered. So there's basically the naked DNA has entered inside these cells. Okay, and then they have incorporated themselves. So basically these are recipient cells or the competent cells. So now let's talk about what is competence. Competence is the ability of a recipient bacteria to take up free DNA and become transformed. It's called competence. Okay, it is an inheritable character. So these are called competent. So basically all the cells are not competent. Okay, some are competent, some are non-competent. Competent bacteria that takes up DNA from environment are at high frequency encode protein called as competence factor or competence protein. So basically the cells that have competence protein are competent. Now competence factor facilitate binding and uptake of DNA. Now, without this particular factor, a cell cannot take up DNA. Now, let's see how this particular things work. Okay, now let's see how this competence factor works. So, competent bacteria, I have drawn a diagram of a competent bacteria. And out here you can see there are some yellow proteins. So, these are the competence factor which is made by this particular bacteria, this competent bacteria. Okay, it has made this yellow protein which is the competent factor, and they get incorporated within the cell membrane. So, through this cell membrane, what will happen? All the DNA can now enter. Okay. So competence bacteria, they have this competence factor, and what is the function? Binding foreign DNA to cell surface and uptake by cytoplasm. So without this competence factor, that means this yellow protein, this competence factor, without this, a cell cannot become competent. And if a cell is not competent, then they will not be able to take up DNA. So there will be no transformation. Okay, so in order for transformation, the cell has to be competent. For that, they require this competent factor. Okay. Now let's talk about the process and the steps of transformation. Okay, self-free transforming DNA in the bacterium can be naturally released into the surrounding media when a cell dies and undergo lysis. Once foreign DNA comes in contact with competent bacteria, linear dsDNA, dsDNA means double-stranded DNA, okay, converts into, this one means single-stranded DNA, and one strand is degraded. So what they are saying is they can take up double-stranded DNA. Okay, they will convert into single-stranded DNA, and one strand will be degraded, and the other strand they'll make. Okay, now single-stranded DNA, that is the exogenous. Exogenous means the DNA that is coming from outside. Okay, the foreign DNA is unstable and degraded unless they are integrated into endogenous by homologous recombination. So, in order to transformation to take place, what they have to do, whatever DNA is coming in, they have to take that DNA into the endogenous. Now, what is endogenous? Endogenous means here I am talking about the bacterial chromosome, what they are already having. In that, it has to get incorporated. Okay. So, bacterium cells that take up foreign DNA or transforming DNA are called as transformants. Okay, so this is how we get our transformants. So, I'll explain this with the help of a diagram now. Okay, now let me explain it with the help of a diagram. So, out here, bacteria is going through lysis. So, that means the bacteria is being broken down. And after breaking down, here we are getting like these red ones. These are all double-stranded DNA. Okay, these are all double-stranded DNA fragments from dead cells in the medium. Okay, so in the medium, these are all the dead cells. From the dead cells, we are getting this double-stranded DNA. So these are all the double-stranded DNA. This one is also the same thing. Okay. Now, in the next one, I have drawn some green cells and some purple cells. The green cells, these ones, they are the competent bacteria. So these are the competent bacteria. That means the bacteria that can take up this double-stranded DNA. And the purple ones are the non-competent bacteria. That means they cannot take up DNA, and both of them are recipient. Okay, even though they are recipients, only the competent ones can take. Okay. Okay. Next. Okay, next what happens? Now, the competence bacteria, whatever bacteria was competent, only those bacteria will be able to produce the protein, the yellow protein that I was talking about, that is the, so this one is the competent bacteria. Okay, and this competent bacteria, they are making this competent factor or competent protein. Okay, so this is the competent factor or protein. Okay, they are making, and on all of these are going and getting incorporated within the cell membrane. Okay, so now what will happen after this is that the DNA can now enter, and whatever this one is the non-competent one, non-competent bacteria. Okay, so there is no competence factor. Okay, all right, next what is happening, this double-stranded DNA. Okay, so again, this one is also the double-stranded DNA from the medium. Okay, the same thing, which I had written out here. The same foreign DNA. Okay, this is also the same thing. This is also the same thing. Okay, now this thing will go and enter inside the cell. Okay, within the competent cell. So what will happen? These double-stranded DNA out here, you can see it was double-stranded. Now it's becoming single-stranded and it is entering. Only one of the strand will enter inside the cell, as you can see in this diagram. It was double stranded previously, but only one strand will enter, and the rest, whatever DNA is present outside, they are all double stranded. Okay, they are all double stranded, but only one single strand will enter inside. Okay, so I'll just write that down. So basically what's happening is only single stranded DNA, that means it's the foreign DNA, enters the cell. ठीक है, only the foreign DNA is entering inside the cell. Now next step. Okay, okay, so what's happening out here now in this particular step, what we are seeing is there is homologous recombination taking place. Recombination. Okay, out here homologous recombination is taking place. Where? In this one, in this particular bacteria, homologous recombination taking place, not in the other one. Okay, we'll see how. So in this particular bacteria, we can see that out here this one is the bacterial chromosome. Okay, this one is the bacterial chromosome, and you can see some of the red part is also there. So plus we also have the exogenote, or you can say the foreign DNA. So both the parts are present out here. Okay, so basically what's happening, this extra whatever DNA had entered, it is now getting incorporated. Okay, so now I can, so now this one is a transformant, and the other cell out here, again I have this foreign DNA. So out here what's happening is the foreign DNA is being broken down. Okay, so we can say depleted foreign DNA. So the foreign DNA is getting depleted and nothing is getting incorporated. So nothing is getting incorporated out here. Okay, and therefore the cell will remain a non-transformant. So out here we can understand, in order for transformation to take place, is very very important for a cell to be competent, and then only it will take up DNA from the external environment, that is the matrix, and they will become a transformants, and this is the process of transformation. Now finally let's talk about some of the importance of transformation. So what is the importance of transformation? Now, transformation is used for gene mapping. Okay? Genetic analysis of transformation is based on principle that two marker gene transfer together if they are near enough to be carried on the same DNA. So that means that if two of the DNA are together, like very close together, then they'll be passed on together. So that's how it helps in gene mapping and marking. Okay, where the genes are present. And the other is frequency of co-transformation is inversely proportional to the distance between the gene. So it can also help you to understand the distance between genes. Okay, so this is how it helps. So that's all in this particular video. If you have any doubt, you can ask me.